Welcome to this special episode on systems medicine and organ crosstalk. This is a journey into how the body really works when we stop thinking in silos and start seeing a living network. Today's conversation is based on a 2026 review article in Signal Transduction and Targeted Therapy titled Organ Crosstalk: Molecular Mechanisms, Biological Functions, and Therapeutic Interventions for Diseases by Che and colleagues. It is a dense, multi-layered paper. What you're going to hear now is that science translated into a 20-minute story you can listen to on a walk, in the car, or between rounds. Let's begin. Imagine zooming out from a single organ, the heart, for example, and instead seeing the entire body as a shimmering web of connections. The brain sends autonomic signals to the heart. The gut microbiome sends metabolites to the liver. The kidneys adjust hormones that reshape the vasculature. The lungs modulate immune tone. Adipose tissue talks back to the brain about energy status. This is organ crosstalk, also called organ axis or organ interaction networks. Traditionally, medicine has been organized organ by organ: cardiology, hepatology, nephrology, neurology. That has produced great depth, but it misses the dynamic feedback loops between organs. The review you've just heard excerpts from systematically maps these loops and then asks, if disease is networked, shouldn't diagnostics and therapy be networked too? Che and colleagues searched the literature from 2015 to 2025 using organ pairs and keywords like crosstalk, axis, and network, and identified over 50,000 relevant articles. When they plotted this as a Sankey diagram, three hubs emerged very clearly: the brain, the gut, and the liver, with highly studied pairs like brain, gut, liver, and brain, heart. At the same time, many organ pairs like stomach, bone, or spleen, adipose are barely explored. So the field is rich but uneven. Some axes are well mapped, others are almost blank. To make this knowable in audio form, let's structure the next 15 minutes into 3 acts, the 4 super systems that mediate crosstalk. Key organ axis: brain, gut, brain, liver, brain, heart, and the heart-centered lung, liver, kidney, and gut axis. Technologies and therapies: how to actually use this network view at the bedside. ACT1, the 4 super systems. The first big idea from this review is that almost all organ crosstalk runs through 4 overlapping systems: the circulatory, nervous, endocrine, and immune systems. The circulation is obvious. Blood flow delivers oxygen, nutrients, hormones, metabolites, and inflammatory mediators between organs. If you reduce cardiac output, the kidney and brain suffer. If you block portal flow, the gut and liver both destabilize. The nervous system provides rapid electrical communication. The vagus nerve, sympathetic fibers, and enteric neurons are constantly sending status updates: how distended the gut is, how full the lungs are, how high the blood pressure is. The review highlights vagal circuits connecting gut, liver, and brain as critical for homeostasis and, when dysregulated, for disease. The endocrine system transmits slower hormone-based signals: insulin, cortisol, natriuretic peptides, thyroid hormone, growth hormone, that simultaneously affect multiple organs. Think of insulin produced by the pancreas acting on muscle, liver, adipose, and brain, or cortisol, a stress hormone that reshapes immunity, metabolism, and vascular tone all at once. And finally, the immune system, both innate and adaptive, links organs through cytokines, chemokines, and mobile immune cells. A sterile injury in one organ, like myocardial infarction, can trigger systemic inflammation leading to acute lung injury, kidney dysfunction, or neuroinflammation. So when we say heart, kidney axis, or gut, lung axis, what we really mean is these 2 organs are talking through this 4-channel communication infrastructure. With that in mind, let's go axis by axis. ACT2, brain-centered axis. The brain is described in the review as both conductor and recipient. It sends signals to peripheral organs and also reshapes its own function based on feedback from them. 3 axes are highlighted: brain, gut, brain, liver, and brain, heart. First, the brain-gut axis. Here, 3 pathways dominate: neural, neuroendocrine, and immune. Neurally, the vagus nerve carries signals in both directions. Gut distension, nutrient content, and microbial metabolites all influence vagal afferents. In response, efferent vagal fibers modulate motility, secretion, and even fat absorption through brainstem nuclei like the dorsal motor nucleus of the vagus. Neuroendocrine signaling is mediated largely through the hypothalamic pituitary adrenal axis. Stress activates hypothalamic CRH, then pituitary ACTH, and finally adrenal cortisol release. Cortisol slows gut motility, damages epithelial barrier integrity, and triggers pro-inflammatory cytokine release in the intestine, such as TNF-alpha, IFN, and IL-6. Those same cytokines can feed back to the brain, sustaining HPA activation and altering serotonin pathways and brain-derived neurotrophic factor, reinforcing a loop between chronic stress, depression, and inflammatory bowel disease. Immune pathways add another layer. The gut microbiota continuously stimulates innate immune sensors and inflammasomes. Dysregulation of these pathways is linked to neuroinflammatory conditions like multiple sclerosis, Alzheimer's disease, Parkinson's disease, postpartum depression, and neuropsychiatric symptoms more broadly. In both animal and human studies, intestinal dysbiosis and increased gut permeability correlate with cognitive decline, autism spectrum traits, and mood disorders. Therapeutically, the brain-gut axis can be modulated through probiotics and prebiotics, which restore microbial balance and increase short-chain fatty acid production. Through psychotropic drugs like SSRIs that work on both central serotonin and gut motility, through vagus nerve stimulation, dietary interventions such as Mediterranean and MIND diets, and even fecal microbiota transplantation in selected conditions. Second, the brain-liver axis. Here, the key roles are metabolic regulation and inflammation. The sympathetic nervous system via splanchnic nerves releases norepinephrine onto hepatic receptors, promoting glycogen breakdown, gluconeogenesis, and inflammatory activation. The parasympathetic vagus, by contrast, favors glucogenesis and reduced hepatic glucose output. Brainstem and hypothalamic circuits, including AGRP neurons, sense energy states and adjust hepatic autophagy and metabolic remodeling via stress hormones such as corticosterone. The liver sends signals back to the brain through bile acids, cholesterol, lysophospholipids like LPA, ketone bodies, and cytokines. Bile acids, for example, can modulate brain function directly or via gut-brain pathways, while hepatic-soluble epoxide hydrolase activity influences amyloid metabolism and Alzheimer's disease pathogenesis in mouse models. Exercise-induced hepatic factors like GPLD1 have been shown to improve cognitive function and neurogenesis in aged mice. Dysfunction in this axis contributes to hepatic encephalopathy, where accumulated neurotoxins such as ammonia alter brain function, to neurodegenerative disease where disturbed bile acid and cholesterol metabolism interacts with amyloid processing, and to metabolic disease where insulin resistance and obesity reflect impaired brain-liver communication. Emerging therapies here include TGR5 agonists, SEH inhibitors, ketogenic diets, gene therapies targeting liver enzymes like FS2, and immune modulation such as blocking monocyte infiltration into the brain during liver inflammation. Third, the brain-heart axis. This axis is dominated by the autonomic nervous system, the HPA and SAM stress systems, and neuroimmune coupling. Sympathetic activation via norepinephrine accelerates heart rate and contractility through adrenergic receptors, increasing cardiac output in acute stress but promoting arrhythmia, hypertrophy, and fibrosis when chronically overactive. Parasympathetic vagal tone slows the heart and is protective. Chronic disruption of this balance links psychological stress, PTSD, depression, and anxiety to higher cardiovascular risk. Through the HPA axis and cortisol, chronic stress also promotes sodium and water retention, hypertension, dyslipidemia, and central adiposity, all risk factors for heart failure and coronary disease. The sympathetic adrenal medullary system's catecholamine surges contribute to Takotsubo cardiomyopathy and post-stroke cardiac dysfunction. Inflammation bridges the organs. Sympathetic activation of lymphoid tissues mobilizes monocytes and neutrophils, which secrete cytokines like IL-6 and TNF-alpha, driving myocardial inflammation and remodeling. Cardiac cytokines and DAMPs then cross into the brain and activate microglia, contributing to depression and cognitive decline. Interventions on this axis include vagus nerve stimulation, ACE inhibitors and ARBs, beta blockers, SGL2 inhibitors with neuroprotective signals, and lifestyle measures like exercise and stress reduction. So from the brain's perspective, health is constant negotiation with the gut, liver and heart via neural, hormonal, immune and metabolic languages. ACT3 heart, lung, liver, kidney and gut-centered axis. The review then turns to the heart as a hub, and from there explores heart, kidney, heart, lung and heart-liver crosstalk. The heart-kidney axis cardio-renal crosstalk is built around hemodynamics, RAAS, SNS activation and inflammation. In heart failure, reduced cardiac output lowers renal perfusion pressure. The kidney senses this via the juxtaglomerular apparatus, releases renin and activates RAAS, causing vasoconstriction, sodium and water retention, and rising blood pressure and congestion. RAS overactivation promotes myocardial hypertrophy, fibrosis and progressive HF, while renal venous congestion angiotensin-mediated microvascular changes reduce GFR and push the kidney toward acute or chronic injury. Conversely, chronic kidney disease and AKI generate uremic toxins, oxidative stress, and inflammatory mediators that induce left ventricular hypertrophy, diastolic dysfunction, arrhythmias, and accelerated coronary disease. Electrolyte shifts, notably hyperkalemia, further destabilize cardiac conduction. The therapeutic palette for cardiorenal crosstalk includes ACE inhibitors, ARBs, mineralocorticoid receptor antagonists, SGLT2 inhibitors, IL-1 beta blockers, antioxidants like N-acetylcysteine, beta blockers, sodium restriction, and structured physical activity. The heart-lung axis is about coupling pump and gas exchange. Left-sided heart failure raises pulmonary venous pressure, causing interstitial edema and impaired oxygen diffusion. Stiff ventricles in HFpEF create exertional dyspnea via VQ mismatch and increased pulmonary pressures. On the right side, chronic lung disease, COPD, pulmonary fibrosis, pulmonary embolism raises pulmonary vascular resistance, straining the right ventricle and eventually leading to COR pulmonale. Inflammation and hypoxia play key roles. Myocardial injury can trigger ARDS like lung damage. Lung inflammation, as in severe pneumonia or COVID-19, releases cytokines and ROS that injure the heart and vasculature. Obstructive sleep apnea with intermittent hypoxia and intrathoracic pressure swings is particularly harmful for both organs via SNS and IAS activation. Therapies include beta blockers, optimized mechanical ventilation strategies, IL-6 blockers and corticosteroids in ARDS, pulmonary vasodilators like prostacyclin analogs and PDE5 inhibitors, and lifestyle interventions like smoking cessation and pulmonary rehabilitation. The heart-liver axis closes the cardiocentric triad. Here, congestive hepatopathy and cardiohepatic syndrome are central. In heart failure and pulmonary hypertension, elevated central venous pressure is transmitted to hepatic veins, causing sinusoidal congestion, centrolobular necrosis, and eventually fibrosis and cirrhosis-like changes. Acute low-flow states like cardiogenic shock can produce hypoxic hepatitis with massive aminotransferase rises. On the flip side, chronic liver disease alters hemodynamics and metabolism in ways that stress the heart. Cirrhosis leads to high cardiac output but blunted contractile reserve, diastolic dysfunction, and arrhythmia vulnerability, collectively termed cirrhotic cardiomyopathy. Metabolic associated fatty liver disease and NASH promote coronary atherosclerosis and HF via shared insulin resistance, dyslipidemia, and inflammation. Therapies that straddle this axis include non-selective beta blockers to reduce both portal pressure and sympathetic drive, GLP-1 receptor agonists and SGL2 inhibitors to improve metabolic and hepatic profiles while protecting the heart, mineralocorticoid antagonists that signal via FGF21 to aid post-infarct remodeling, and dietary and exercise interventions like the Mediterranean diet and structured physical activity. From the liver's perspective, the pivotal axis is gut-liver. The review elegantly illustrates how in health bile acids, phospholipids, bilirubin and cholesterol are secreted into the gut where they shape the microbiota. In return, microbial fermentation produces SCFAs, vitamins, secondary bile acids and other metabolites that travel back to the liver via the portal vein. In disease, dysbiosis leads to increased flux of LPS, ethanol from endogenous alcohol-producing bacteria, TMAO and other toxins. activating Kupffer cells, stellate cells, and inflammatory pathways that drive MAFLD, NASH, ALD, cholestatic disease, and HCC. Altered bile acid signaling via FXR and TGR5 further undermines barrier integrity and metabolic control. Interventions target both ends, probiotics and prebiotics, FXR agonists like obeticholic acid, TLR4 inhibitors, phage therapy against high alcohol producing Klebsiella FMT, and high fiber Mediterranean style diets. The kidney's main gut partner is captured in the gut-kidney axis. Here, beneficial metabolites such as SCFAs and vitamin K protect the kidney, while toxic products like presyl sulfate, indoxyl sulfate, IAA, and TMAO drive CKD progression, cardiorenal disease, and mortality. CKD in turn alters the microbiome, weakens gut barrier function, and increases systemic endotoxemia. Again, therapeutic levers include probiotics, prebiotics, diet, microbiota-targeted drugs, and FMT. Finally, there is the gut-lung axis. SCFAs and tryptophan metabolites modulate lung immune cells, dampening allergic inflammation and enhancing antiviral defenses, while dysbiosis and harmful metabolites like TMAO and succinate predispose to asthma, COPD, ARDS, pulmonary hypertension, and worse outcomes in viral pneumonias. Lung injury feeds back to the gut, altering microbiota composition and increasing permeability, which amplifies systemic inflammation and organ damage. Probiotics, high-fiber diets, FMT, and specific pathway inhibitors are all under investigation to tune this axis. ACT4 methods, technology, and the future. The last part of the review steps back and asks, how do we actually study and apply organ crosstalk? For technological pillars are described imaging, experimental models, molecular and omics approaches, and artificial intelligence. Multi-organ MRI, CT, PET, and SPECT can now map anatomy and metabolism across heart, brain, liver, and kidneys in the same individuals. Data from tens of thousands of UK Biobank participants reveal correlated patterns of liver fat, cardiac function, aortic stiffness, and brain structure, the so-called heart-brain-liver axis. Disease models from AKI to stroke to sepsis to COVID-19 have been used to dissect how injury in one organ propagates to others. Animal experiments allow targeted manipulation of pathways like IL-33ST2 in cardiorenal syndrome or vagal circuits in gut-brain communication. Organoid and organ-on-chip systems now model the liver, islet axis, heart, kidney axis, and multi-organ responses to stress like cosmic radiation, providing controllable platforms for drug testing and mechanistic work. Molecular and omics-level studies, metabolomics, proteomics, transcriptomics, epigenomics reconstruct how metabolites, hormones, cytokines, and epigenetic marks transmit signals between organs. Examples include bilirubin and gut microbiota in liver, gut signaling, or SCFAs and neuroactive metabolites in the gut-brain axis. AI ties it together. Deep learning on imaging predicts inter-organ dysfunction from subtle patterns. Machine learning on EHRs and multi-omics data identifies new organ network phenotypes in heart failure, cancer, and kidney disease and discovers drug-omics relationships for repositioning. The authors end with a call for network-driven models and a move toward systemic personalized medicine. Instead of managing heart failure, NAFLD, COPD, CKD as separate diagnoses, they argue for building multi-organ risk scores, tracking axes like gut, liver, kidney or brain, heart, lung, and designing therapies that act on multiple organs and pathways simultaneously. They also highlight collaborative megaprojects like the Human Phenome Project and Digital Human initiatives, aiming to build a truly integrated organ network map. And they point out that much of today's literature is still organ-centric. Network analyses are only beginning. As you leave this episode, you can think of every clinic patient in front of you differently. The HFpF patient is not just a stiff left ventricle, but also a congested liver, a dysbiotic gut, a stressed brain, a vulnerable kidney. The MASLD patient is not just a fatty liver, but a brain, liver, heart, gut network under metabolic and inflammatory strain. For clinicians and researchers, the message from this review is clear. Understand the axis, treat along the axis, and design trials that measure multi-organ outcomes. For educators, it suggests a new curriculum, less systems in isolation, more systems in conversation. And for integrative preventive medicine, it provides a rigorous mechanistic backbone. Diet, exercise, stress management, sleep, environment, microbiome, and targeted drugs are not alternative, but key levers on a complex organ network. Thank you for listening to this 20-minute tour of Organ Crosstalk. The original review is open access in Signal Transduction and Targeted Therapy 2026, Volume 11, Article 8, if you wish to dive into the full diagrams and references yourself.